Hello, everyone, and welcome to our monthly Science of Alt Protein seminar. I am so excited to be welcoming you all into this space. My name is Amy Huang, and I'm the Associate Director of Scientific Ecosystems here at the Good Food Institute. For those of you who are here for the very first time, welcome. The Good Food Institute is a nonprofit think tank that is 100% driven by philanthropy, and our goal is to build a world where alternative proteins are no longer alternative. If you're looking to take a deeper dive into the science of plant-based fermentation or cultivated meat technologies, check out our open access resources at gfi.org/science. Today, it's my pleasure to introduce Dr. Fabiana Bressan, a GFI grantee and associate professor at the Veterinary Medicine Department at the University of São Paulo. Fabiana graduated from the Veterinary Medicine program at the University of São Paulo in 2005. She then went on to earn her Master's in Science degree in 2008 and her PhD in 2013. And her research is very much focused on cell reprogramming by gene induction to achieve pluripotency. She also studied nuclear transfer and stem cells in human and animal models, and Fabiana completed her postdoctoral studies in stem cells and cell therapy. At the University of São Paulo, she currently focuses on the study of in vitro generation of primordial germ cells, gametes, and epigenetic reprogramming. During today's seminar, Dr. Bressan will describe the potential of induced pluripotent stem cells generated through cellular reprogramming for cultivated meat production. Her talk will address both the stem cell biology and the generation of iPSCs, relevant applications, and recent advancements in the field. So with that, I am honored to welcome Dr. Bressan to the stage to share her insights with us. And Fabiana, please feel free to share your screen, take it away whenever you're ready. Awesome. Thank you so much, everyone. And over to Fabiana.
Thank you very much, Amy. Thank you very much for the invitation. It's a pleasure to be here. So thank you, Amy. Thank you, Henny. I would like also to thank the GFI Brazil, who is always giving us great support here in our research. So thank you, Amanda and Luciana and many others. So thank you, everyone who's here today. Maybe this is a different webinar from the ones that I have seen from GFI. It is very focused on cell biology, and I hope we get at the end with a very open mind, possibilities to use this new technology, which are this pluripotent stem cells. So I'd like to start showing you our university here in São Paulo, Brazil. This is our main building. And yes, it looks like a farm because we have the pleasure to be in a university which was formerly was a farm. So we have here 4 courses: veterinary medicine, animal sciences, food engineering, and biosystems engineer. And all of our students and people then, they are always looking for a better use of our animals and to result in a better world, thinking about sustainable food. This is our university, just to have a quick look at this and to show you where we are. We are at a farm, so yeah, we look forward to have a good use of our animals and to have food from them, but of course in a very sustainable manner. From our specific group, I would like to share our general aims, which are very general but ambitious. They are to contribute to regenerative medicine and show you how, to the conservation and propagation of genetic material. We are in a vet med school, and of course to animal and food prediction. This may seem a little bit overwhelming, but we have a very recent and robust new technology to help us to achieve these goals, which is to use specifically the cellular reprogramming, which is something we are going to talk a lot today, to generate
viable and robust cellular models, functional tissues, products from these cells or tissues. And these cells and these tissues, they are derived from animal cells, thinking with the greatest aim to avoid to maybe to diminish their use as models or as food. So we can do that today. We can have lots of different cells and systems and tissues that works in vitro, and I will try to show you how we can do that. And the keywords of this talk, I would like them to be non-invasive and in vitro in a sustainable manner to try to optimize the use of these animals at the most. So we are doing the cellular reprogramming first in recent years, mostly aiming regenerative medicine. So we were able to model several diseases, both for veterinary and human medicine, and I will show you some examples. More specifically, we has been able to produce primordial germ cells, which are the gamete precursors in vitro. And these cells, they are very differentiated, very specific. They are probably the most difficult cells to achieve in vitro. And as long as we are getting success in this part of the science, I would say, so the next step, we thought, why not go directly to the food chain production? So we are working on this, on both of these subjects at this moment, on reproductive medicine and also on cultivated meat. And this, we already have some preliminary results I would like to show. I would like also to show both sides, the regenerative medicine, what we have from applications of the cells, and also the cultivated meat in this. First of all, we have to understand how to produce, how to generate these cells that I am telling you that they have so many applications. And to start thinking about how to produce these cell lines, it's a fundamental question for us to understand what is happening on the normal development of an organism. So this is a very beautiful drawing from Professor Paul Hutto, a former collaborator of our group. And here he shows that once an oocyte is fertilized by a sperm, the genetic material of this new organism, it maintains the same until the end of its life. But the cells, they are dividing, they are compromising, they are getting more specialties, more specific morphology and function, and it does not have to do anything with genetic alterations. It's our epigenetic. We will try to understand what it is. So this is a first question. We have to think about what is happening here after we have one embryo cell, and after some divisions, we already have more than 220 types of different cells in one organism. And if we can understand each part of the cycle, maybe we can modulate it and make the decision, the fate decision of each cell that we want them to be. So that's the central point of this talk. And this is a very nice representation of a node theory. It's called Waddington's epigenetic landscape. It was proposed by this researcher. It's not very recent. What it says is that whenever we have an early embryo, it's recently fertilized, this embryo can choose his pathways during his development. So as long as the cells are dividing and they are choosing, or they are being affected by the environment and going through hills in a mountain, and these hills, as these cells go further in development, they acquire specific marks, specific genetic marks, then makes the cell lose their potency, However, they acquire a specific phenotype and specific function. Today, we know which are these specific epigenetic marks that each of these potential states of the cells have. And as far as we know what we are seeing, we are able to change it. So here are some initial concepts that I would just like to remember everyone, so everyone is at the same base. A totipotent cell is that very early embryo that has no cellular differentiation, has not activated his own genome yet, so it's a very rare, it's very difficult to deal with, and we currently do not have this kind of culture in vitro. The unipotent cells on the other sides are that those cells that are already very specialized and differentiated, and they do not replicate well. So for cellular therapies or for any other use in large scale, they are not our targets, but I will show you how they can recently be used. So in this talk, we'll just have a very brief glimpse on this other type of cells, the pluripotent cells, the multipotent cells, and then how we can use already differentiated cell types from adult organisms in order to produce our large-scale cell-derived products. So just to remember, the pluripotent cells are those who were previously isolated from embryos in the blastocyst stage from the inner cell mass. This inner cell mass and the blastocyst, they are responsible for forming all of our adult tissues. So we can, at the time of the embryo at blastocyst stage, it would be something like day 5 to day 7 in humans or bovine, for example. It's species specific. We can maintain them in culture and use them to differentiate into virtually any other type of tissues of an organism. This was at the time, 80s or 90s, considered as a holy grail of the regenerative medicine because it virtually cures every syndrome or every condition that needs new cells or functional tissue. We are going to see that they also have a lot of problems, and that's why we don't use them much nowadays. On the other side, we have those cells who are already a little bit more advanced in their development. They are multipotent, mostly from adult tissues, but also from fetal or newborn. Almost every tissue of our organism has some part of the cells, a few number of the cells which are a little less differentiated, and it's less differentiated they are, more replicating they are. In some tissues, we have the progenitor cells, which are the cells, they do not differentiate much, they still can divide into more specialized cells. The cells, they are very difficult to isolate and maintain in culture. In some tissues, the isolation of these multipotent cells is not recent, and we have several cases of success, thinking on therapies, and the transplantation of the bone marrow is one that has saved millions of lives, and we know exactly how to do that and which cells we are isolating, how we can do that, and what to expect. So not every other tissue has this facility to isolate the cells and to replicate the cells. So we can have mesenchymal or multipotent stem cells from adipose tissue, from the dental pulp, from hair follicles. These are the ones that we are used to cure, and they are already on the use of the medicine. It's nice to remember that a few decades ago, or even years, we used to believe that these multipotent cells, they are best characteristic was to differentiate into some other cells. They do not have the same potential as the embryonic stem cells. They cannot be differentiated into any other type of cells, but yes, they have this potential of still differentiate or specialize in one or two or three functions. But this is very rare. This is in a very small scale. It's not every tissue that we can recover a great amount of the cells. So we know now that the specific characteristic that make the mesenchymal or multipotent stem cells a very nice technology to be used in cellular therapies is because they secrete a lot of things that helps the immunomodulation of tissues. They are anti-scarring, they are anti-apoptosis, they also can modulate angiogenesis. So probably these cells will be continue to be used for a long time in medicine, but not because they differentiate or because they have a long life in culture, but because they will be used as drugs, as pharmacy. And this is a very nice use of these cells. So both of these cells, which we are talking about, pluripotent or adult cells, they have important limitations when we think for any purpose that will demand a large-scale culture. So the pluripotent cells, of course, the simple isolation of the cells from an embryo results in a destroyed embryo, so it has very important implications. Also, we do not actually know exactly this genome, the genetic material, because of course every embryo will be unique, and this is very important. So it limits its use both for cellular therapies, for medicine, and for any other application. And the adult cells, as we have already talked, they are very limited. Their isolation often needs invasive procedures like biopsies, and the amount of the cells that we recover are usually not enough for our large-scale applications. So we have to have a solution for this if we want to do some tissue or some cell culture in large scale feasible. And the pluripotent stem cells, they have one specific point that is very important if we are thinking about cultivated meat, is because the maintenance of pluripotency of the cells in vitro, when we derive these cells from farm animals or domestic animals or any animal apart from humans and mice, is still not well reported. So there are much work to be done in this sense yet. They are they are not adequate for food production. So then we come back to our basic sciences, and then I start understand what I will propose to you in this talk. So if we know what makes a cell differentiate and commit into a specific morphology and function, and it is related to its epigenetic status, which is the same as the cellular, its gene expression profile is changing during its development. So if we can modulate it, we will be able to get a cell from an already adult organism and reprogram the cell into the pluripotent state again. So it is not done commonly in the nature unless we are talking of germinative cells and gametes. So this coming back of the fate of a cell, what we call cellular reprogramming, because we get the DNA of the cell, the genetic material, and we reprogram this in order to show the cell. So you are expressing genes A, B, C, but I want you to stop expressing these genes and start expressing genes M, N, and O. So if we can do that with the cell, so virtually, if we have one cell, we can generate any other cell in one organism. It has been proved correct, and most of you may remember Dolly, who was a sheep that was cloned. So the process of cloning or somatic cell nuclear cloning is one technique that relies on cellular reprogramming. So I'm sorry, this is in Portuguese, but I can't translate it. We have one animal and one cell from one animal, virtually any cell from that animal that has a complete material, genetic material. And I can put this genetic materials inside of an oocyte, a female gamete, we have already taken its own genetic material off. And this oocyte understands that he can start a new life, and he reprograms, and this oocyte reprograms the adult cell into a pluripotent state. This is what we call somatic cell nuclear reprogramming. So this new embryo, it starts to divide and it starts to go down that hill all over again. And this technique has been already very proved, is already very proved. It's a robust. Here's just some examples of animals that were born, that were cloned, and you can see that the time that they were born, it has been already some years, some decades, so it's not recent. And it's something that we are trying to still understand is which specific factors that the oocyte has that we could use to do that in vitro. And some years ago, far more recent, some researchers did some extraordinary work trying several different profile gene expressions to make the same process of cellular reprogramming, however, all in vitro. And how they did that? So they had their initial primary lineages of fibroblasts, and they overexpressed, they inserted exogenous genes in several combinations in these cells, and they came through very specific group of genes that were able together to reprogram the cells the same way the oocyte did. So this was called induced pluripotency, or in vitro induced pluripotency, and these cells are called iPS cells, or induced pluripotent stem cells. And this was very important to our science community, because for the first time, they showed that it was possible to overcome all those epigenetic programming without the use of the oocyte and without creating an embryo. So this, both techniques, nuclear transfer and induced pluripotency, they were awarded the Nobel Prize in 2012 for the discovery that mature or already specialized cells, already differentiated cells, can be reprogrammed to become pluripotent again if we know what the code is of this programming. So this was like the new holy grail for therapies, for cellular therapies, because now we are able to get a cell from any kind of organism, to culture the cells in vitro, to induce the cells, reprogram, and in approximately 20 to 30 days, we have a very open and it's like an embryonic stem cells, but without the embryo without destroying an embryo. And most importantly, as I have already told you, we do not have robust pluripotent cells from embryos when we are talking about farm animals or domestic animals. But now we have this protocol so robust, although we have to have some optimization between species, but it's very robust, and we can very conservative this reprogramming. So we now have real pluripotent stem cells from other animals. So what is happening here? So as soon as we make these cells that already has a memory, for example, the cells that came from a skin of a cow, as soon as we make the cells express again a group of specific factors, usually transcription factors. These are the most common of 4, KLF4, SOX2, and SEMIC, but we have now several other combinations that we know that works. These cells, they start forgetting all the marks that the memory that these cells have from the skin, and they are being prepared to have a very open chromatin to receive other programming. For example, it would be very easy for us to differentiate the cells in any other cell type as long as we have the specific protocol for that. So of course, if we want to produce a muscle cell line, it's very difficult to have direct differentiation from a skin cell for muscle cells. But if we can reprogram the cells and make them forget they were on skin cells, they can be modulated to have the muscle morphology and function. So this was actually a very nice novelty for medicine, and also for other applications, as I will try to show you. So, okay, how we do that? So, if we want to have this very open and very pluripotent stem cells, we have to first have the primary cells. These primary cells can be virtually any other type. So it's very usual to have biopsies from skin or from fat tissues, but I will show you that we can have the cells from tissues just like urine, a blood collection, saliva, from hair follicle, which are really non-invasive, and we do need very few cells to start the process. If we have, for example, 1,000 cells, for sure, if we have a very optimized protocol, we will have then millions or billions of iPSCs after reprogramming. So we have our initial cells, fibroblasts, urine cells, cells from milk, whatever cells you have from that organism that you want the stem cells. We do the reprogramming, so by putting the exogenous factors working inside the cells. After some days, we transfer the cells for a very non-differentiating environment. So it's the same media used for pluripotent cells that came from embryos. And then approximately in less than 1 month, we have millions and billions of pluripotent stem cells that are ready to be differentiated in any tissue that you want. So, of course, the process is not 100% efficient. This is an example on the right of a very beautiful iPSC colony. It resembles very much the embryonic stem cells, and on the left, some cells that started the reprogramming process and failed during this time. So reasons for such failure: yes, the species matter, the animal age. The animals that are younger, they usually reprogram better. The cell passage number matters. So cells that are freshly isolated or just went through cultures for a few passages, they are easily reprogrammed. The cell type, of course, so it's obviously easier to reprogram a more undifferentiated cells than a more specialized cells. So it's easier to reprogram a fibroblast than a muscle cell or a neuron. The choice of the vectors that we use and the method of reprogramming has some, it is important for the efficiency and also the supplementation conditions that are more prone to be different between species. So probably species, we use, for example, FGF for humans and for cattle, and we use leaf for maintaining pluripotency in mouse and in dogs. So in other animals such as domestic, wild, farm animals, we will have to study what we have as background information on pluripotency to be sure that we have a very nice pluripotent colony. So this is one important question, one important point to be very clear. We have already said that the adults themselves, they have their potential, but it's very limited, and they will not solve the problem of a large-scale cellular prediction. The embryonic stem cells would have this potential. However, there are very important ethical and other issues applied to them. And first of it would be the embryo, the law that we lose the embryo, it is actually destroyed. And also the genetic background from the cells, they are usually not so easy to study before using. So the induced pluripotent stem cells, they came as the game changer for all those applications that we were waiting the embryonic stem cells to be the holy grail. So because now we have pluripotent stem cells, they are all made in vitro, we can test them, we can know exactly from which genetic background they came. We can, there's no embryo involved, there's no immune rejection involved. We can biobank the cells, and we can have the cells, the isolation of the cells, the primary cells one time, and we can biobank the cells for almost eternity, because once they are pluripotent, they are very self-renewable, and we have the cells in very large quantities. So, okay, let's go to the last part of this talk. Where do we stand on this iPSC? So, are we far from reality? Do we have them now? Is this speculative? What is happening on this field? So, I will show you what we have on our lab today. And then we can, after the talk, maybe discuss a little further on other experiments. So now we have on our lab a biobank. It's called reprogramming cell. We start on our usual models. So it always works with humans and mouse first. So we have the cells. We do not use the cells anymore, but we have them, both primary cells and in iPSCs from them. We have much of our work on cattle and swine for the main reasons that we are in a farm, so we have access to possible and important applications for these animal models. Cattle is very important for food, and the swine also for food, but most importantly, he's a very adequate biomodel for most of human diseases. So we have several studies on these species, but we also have works that have already preliminary results with pets and domestic animals for, of course, other applications rather than food, for example, for cellular therapies. And now we have also a biobank for more difficult species to find. Our objective of biobanking these cells, both primary cells and also pluripotent cells, is to use them mostly on regenerative and reproductive medicine and to propagate this genetic material before they even get to extinction. So yes, we have monkeys, we have eat antlers, we have deers, felines, there's a bat, and a lot of other models. These cells, I would like just to say that they are available for collaborations if anyone has interest on them, both the primary cells and also the already pluripotent cells. So some examples of what we have done with the cells. iPSCs from swine, we have used them to derive neuronal precursor cells. It's very important when we try to model Alzheimer's, but not to only model it in vitro, maybe trying to get it to in vivo treatments. It will be very useful to have a large animal model before going to human themselves. For equine, we have generated several lines of iPS cells from different tissues. We have deeply analyzed them molecularly, I mean, and of course for equine, we pretend to use it in the future, somehow trying to reestablish injuries from those horses that are athletes. These are very interesting experiment, also in collaboration with Professor Christine from Copenhagen. The dog is a very important model for the humans, but he's also a target for us veterinarians. So we have a dysfunction called canine cognitive dysfunction in dogs that should most seen on elderly dogs. It's very similar to Alzheimer's. So we were able to derive iPSCs from young and old dogs, healthy or CCD positive dogs, and next steps include characterizing the cells and trying to come with some kind of treatment, first in vitro and then in vivo. The bovine model was always probably the species that we have more interest in, mostly because our lab has worked before trying to make clones, and it was very inefficient. So the first attempt of our lab to produce bovine iPSCs was actually to use them as nuclear donors to produce clones, because, you know, they were already reprogrammed, already undifferentiated, and we were able to produce some embryos. These are blastocysts from fibroblasts, using cloned blastocysts, using cattle fibroblasts or cattle iPS cells. So we still not sure whether they will give us healthier animals born. We're still on the process of understanding it, but we were able to produce embryos from it. And of course, we have the technology to produce virtually any cell type from this model. One very nice application would be to produce the gametes in vitro. So we have focused most on our bovine and cattle models to try to establish an in vitro gametogenesis assay, of course, to be a part of a program that may not only we could propagate genetic material throughout the world, but also we could try to intervene in some infertilities and to serve also as model for human infertilities. I will show you very briefly these results before going to the cultivated meat itself, because I think it's important for us to have a very broad examples. So what we can actually do with these cells and what has been done already. We used the protocol previously that worked on mice. They used iPS cells and differentiated them into primordial germ cells, and then differentiated these primordial germ cells into viable sperm and oocyte. And these gametes were able to produce viable offspring. This was in mice. We used actually the same protocol that they used in this process to produce the primordial germ cells as any other differentiation, including the muscle cells or fat cells. It's like a cake recipe. If we have the knowledge of what we need to do in each day, we can change the cytokines that we use in order to modulate the gene expression of our cells to our target. So here we used BMP4, stem cell factor, BMP8, EGF in order to have these primordial germ cells, which are very specific cells. And we used this protocol for cattle and for swine, and it worked. So how do we know it worked from using the same protocol? We have already characterized this process. So what we did in a very brief summary, we collected fibroblasts from the skin of some animals. We reprogrammed them using OC4, SOX2, CMYC4, and then we submitted these pluripotent cells to the differentiation, and we analyzed every point, every period, to check what was happening. We had as a result cells that were positive for germinative markers, for both immunohistochemistry and gene expression analysis. When we analyzed it a little further regarding microRNA profile, transcriptome by RNA-seq, and also epigenetic reprogramming of specific genes. And I'm not going to show specific results on this. If someone has any questions on this, we can talk later. And we had the certainty that we were going through the right path. So these cells that we produced, that we thought they were pluripotent, they were really pluripotent, and they were able to differentiate into the germinative cell lineage, which is very difficult to achieve. We have also done part of this characterization in human cells, of course, for aiming a little more personalized medicine. So here we used blood samples collected from women that had a chromosomal abnormality, and from the cells we produced the induced the iPSCs, and then we induced them into germinative cells. And now the next steps, if possible, would be to check whether these cells would give rise to functional and correct gametes for these women to have the chance to produce their own healthy offspring. The perspective of this specific study, the generation of germinative cells and functional gametes, of course, in the veterinary field, it goes further from the therapies. We'd like to, for example, disseminate genetic material from animals that are at risk. For example, we were able to make pluripotent stem cells from the jaguar, and next steps would be to produce gametes from it, and then maybe, who knows, export this material for reproductive programs outside our country. So let's go to the final part of this talk. Let's focus on what we have now and what are the perspectives of these cell lines for cultivated meat. So if we make right now a brief search of what studies have been published on cultivated meat, most of them, and there are lots of them, and even the most recent ones, they still relies on progenitor cells or multipotent stem cells. And as I hope to have shown clearly to you, these are not adequate for a large scale if we want the cultivated meat in our plate anytime soon. So there are several studies on the differentiation process and also on the substrate to make this meat 3D product, and none of them has yet used an adequate source of primary cells. So as a perspective, very recently, there is a revision showing that isolating the cells through biopsies, it's not new, it's a reality. The protocols are well established, the results are here, and okay. And what more? What can we do to optimize this important part of the cultivated meat production? Because if we cannot have a very large scale production of initial cells, we will not ever have a product that is very feasible and adequate consumption or even acceptable. So we have these protocols, they are okay, they are running, but they are based on isolation of multipotent or precursors. So that's a great limitation of the process. As a perspective, recently one group has published cell lineage as a start that is very similar to a pluripotent from the embryo. It's a epiblast stem cell. It's a cell that does not come from the inner cell mass. It's a little bit more differentiated, but it also involves the production of embryos. So it's still not there yet. So what I propose to you, what we have to solve, actually, we have this first problem to solve before solving for which scaffold they must go, which cytokines are best to use or more economically feasible. We have to solve this problem. So the muscle stem cell lines that we are currently using for cultured meat, they cannot be passed in vitro for extended periods. It's a great limitation. So, or we use a cell line that is already limited, or we have to go to embryos. And so what I'm trying to bring to this talks that now have a solution. So we can isolate cells from an adult. We can reprogram the cells, make them self-renewable almost for eternity. We can biobank the cells, and we can then differentiate the cells in a large scale manner. So there are important remarks to be pointed here. The first one, during reprogramming in this process. So most of you must be thinking about a lot of bottlenecks here. So I'll probably be talking about some of them now, very briefly. During the reprogramming and maintenance of pluripotency of these cells, as the cells were first generated aiming cellular therapies, so most of our cellular techniques in vitro, they are already without animal-derived products, which is good, because we are making here a tissue that comes from an animal, but we do not need to use animal-derived products to get to the final product. So we are avoiding the use of these animals throughout this process. Another important remark, I have been talking about skin biopsies, which are most common, and we have seen that multipotent and precursor cells are often isolated after biopsies, but we do not have to use biopsies or punches to have primary cell lines. This is Kayana's work, Kayana Hekia's work, my student. She has generated iPSCs from both cattle and swine, not only sheep, but others in our lab as well, after collection of urine or milk. So it's very, very non-invasive manner to produce cells that can actually be reprogrammed and be used for cell differentiation. So in other words, if you have your cow, which is like a pet for you when you love your cow or you love your mini pig, but you would like to eat beef from it. So can you do that without even touching your animal? So we are proposing that, yes, we can. The third great remark that I would like to show you, that most of technologies nowadays to reprogram the cells relies on producing transgenic cells. It's easier if we integrate the first transcription factors in the cells and then they become pluripotent. It's easier, but it's not the only way. So we have now optimized protocols that we can use in farm animals or domestic animals that do not use viruses or transgenes. So it's not easy, it's not efficient yet. We are working to make that easier for the next studies, but it is possible. So it is possible to collect the cells, even not touching the animal. Yes, we can reprogram the cells without any genetic modifications. Yes, we can maintain the cells without any animal-derived molecule. It's difficult, but we can. So this is what we are proposing now. It's not easy. It's not for this year, but I do believe that in the next year, we can have a whole circle of protocol established for a sustainable large-scale prediction for any functional tissue. And that's include, of course, muscle and fat tissues and weak tissues that you would like to have in your plate. This is a spoiler. We have a ongoing grant together with GFI. We have proposed to make the iPS cells and to make meat, to make muscle cells, to make fat cells, to make some other types of cells in a 3D environment from 2 different fishes. And we have already now actually we have been working with 3 different animals at this species. At this moment, it's going well. I have not brought any more details on that, but I'd be very happy to share some protocols if someone is interested in this proposal. It is still ongoing. This is a fibroblast in vitro culture. We also have some other grants ongoing. They are all ongoing at this moment. They are mostly brought by our governmental funding agencies, so FAPESP and CNPq. And we count on very special collaborations for those. We have been working with the cells that we already have, the iPS cells that we already have from cattle and swine, to differentiate them into different cell types and to generate a 3D structure that resembles the meat itself, the beef itself. And we already have some preliminary results on this. These are very a spoiler. This is really a spoiler. This is also Kayana's work in collaboration with Professor Christine from Denmark. They were able to produce from cattle iPSCs without integration. Now we have several cell lines that are not transgenic. These cells were submitted to differentiation into satellite cells. This experiment is still ongoing, but as a last slide, I would just like to show it that all of we have been talking from the first slide until now, it is feasible, and we have lots of hope on this new technology. This is probably multinucleated cells. We still have to be characterized. So sorry, Kayana, if you're there, I'm spoiling your manuscript. So I just got to the end of this talk just to tell you that thank you very much for all our sponsors, and especially GFI, that made possible for us to have an adequate place to try our reprogramming and differentiation protocols. This is my lab, and these ones that are underlined, they are actually working with the cultivated meat specifically. And of course, we have lots of other collaborators that help us in other disciplines that we do not master. For example, the scaffolds technology. So we join forces to get to a final product. Then I really hope to have shown to you a new technology that is very promising, and I believe it came to solve most of our problems when we think about large-scale production of any functional tissue. So thank you very much for this opportunity. I'm sorry if I talk too fast, and I'm very open to any collaborations or any questions, if any.
No, that was fantastic. Thank you so much, Dr. Bressan, for your presentation. We can feel free to stop screen sharing now, and then move into the Q&A portion. I did also want to signal boost GFI cell line repository for those of you who are doing relevant research and curious about what existing cell lines that are meat relevant are out there. I've just dropped the link there in the chat for those interested. All right, Fabiana, let's move into the Q&A portion. Thank you again for your fantastic presentation. There are a number of questions here that kind of ask a little bit about the selection of cell types. So you had talked about using iPSCs a lot over the course of this presentation. There's a question here about whether you can comment on the relative advantages and disadvantages of using stem cells versus differentiated cells.
Okay, it's a good question, because I don't think we can use actually stem cells like pluripotent stem cells for any applications, because we first need to differentiate them into what we want and then to use it. And if we have an pluripotent stem cells, it just let the cells go to any pathway that the environment tells the cells to go. We can have anything. So if you want to assembly meat, you have to use the stem cells and differentiate them into final fat and blood and conjunctive tissue and muscle cells, so as a final product. So I'm not sure if I answered, but there is not much how to use one or the other. We use the stem cells as a tool to get to our target cells, which will always be the final cells. So the iPS cells, they are just the way we go in a more scalable manner to get to our final target.
Mm-hmm.
Okay.
Thank you. There's another question here about that differentiation step from iPSCs into muscle tissue. Given the challenges you acknowledged associated with that process, what are some strategies that you think could help enhance the efficiency of muscular differentiation? Do you think it might be more effective to use primary myoblasts in the context of cultivated meat production? You did just talk about kind of the scalability of iPSCs, but I'm curious if there is additional context you'd like to add.
It's a very nice question. This is the one I was expecting. So as you could see, I did not included any differentiation protocol in this talk. It was on purpose because they are all still in progress, and we do not have, I do not believe we have one very robust protocol for differentiating iPSCs into our final muscle cells yet, or not, at least for not the species we've been working on. So one question was if it was easier to go from myoblasts to our muscle cells. Yes, for sure. But it does not look the best way to go if you want to think about scalability. So the myoblasts, if you just isolate the satellite cells or the myoblasts, it's a very initial population. First, you need a biopsy. It's kind of invasive. You need to do these biopsies every time. You need to do the procedure again, and the initial population of myoblasts will give you some final cells. If we go for one time getting one iPS cells, as soon as we have this protocol optimized in every step, so we'll need to get the iPS into the myoblast stage and then the myoblast stage in vitro to the satellite cells maybe, and then to go to our final products. We are adding, one step further, but we have billions and billions and billions and billions of cells to try to do that. So we will not even need one biopsy for that. So we can, even for when we try to establish the protocols, and we are trying to do that in several replicates, it's easier to do that on iPS cells than starting on myoblasts, which are, I consider them now as not adequate, but it's a rare source of initial cells to go through all the process. Okay, did I miss anything?
No, I think that was great. Thank you. I will also say that so many of these questions coming through are like entire paragraphs, if not multiple paragraphs. So I think it's safe to say that you had a very engaging presentation. Lots of people from the community who are excited about your research here. My next question is around whether or not you have tried reprogramming on any non-mammalian aquatic animals yet? And if so, what are the major bottlenecks? You obviously touched upon your GFI project that's in the works. Can you speak a little bit more about
Yes, I have not included many spoilers on that. For us, our lab, it's a lot more easier to reprogram mammalian stem cell lines into iPSCs. We have several reprogramming plasmids and viruses that we know which one works for each species. So starting with the fish cell lines was quite challenging. I cannot say much yet because maybe we will have we'll need some patent coming, but we'll probably need optimization not only for the reprogramming process, with the use of very specific fish serum, because our serum may not guarantee the reprogramming process, but also we will try now using fish-specific transcription factors. We were using mouse and humans. We have preliminary results. They are not robust yet to go to the further applications for these iPS cells. So probably we'll go through this change and make new species-specific plasmids for the non-mammalian animals.
Thank you, Fabiana. A follow-up to that is whether you have any plans to work with marine mammals.
I would love to work with them because I would like to have worked with them while I was still on graduation, and I have not had the opportunity to have the cells yet, but I can foresee so many applications for that. So if someone is interested in reprogramming these animals, please come and talk to us.
Yeah, there's a follow-up question to that. Definitely some folks in the community who are interested in this line of work. But looking ahead to that potential work with marine mammals, what problems would you expect to have when working with primary cells and iPSCs? What types of unique challenges might present themselves?
Contamination is one. The other is I'm not sure how to collect the cells in a non-invasive manner because they don't have milk and they don't have hair and they don't have I do not know how we could collect any tissue or fluid without taking them out of the water. That's what we also have struggled with fish. So we had to have the biopsies when we had the fish cell lines. And so this is probably the one first step to think about how to collect the cells. And in case of, for example, one animal has just died, can we collect the cells? So that would be great, for a great use for those animals. And then we could use the cells further. Regarding reprogramming itself, what supplements and what kind of factors they need, I have no idea, because there is not much literature on it. But we can try to reprogram them with what we have already, if that's interesting for someone.
Thank you. There's a question here around whether, kind of relatedly, we need to almost start from scratch every time we're exploring a new animal type in order to develop these protocols, or whether you feel like there are transferable learnings, and that we might eventually emerge with a model that has more extensibility across animal types.
I'm not sure if I understood the question, or if we need to start from the scratch. If we wanna, we just start from the scratch if we want a specific genetic background for a specific application. So if you have a pig that you want stem cells from that animal, or to make an autologous transplant, you have to go from scratch. But of course, if that's not the case, you just want to test something in vitro to make a drug screening, to try some differentiations, and it actually does not mind if it's a female, if it's a bull or a cow. So we can use the cells that are already biobanked, and it's very easy to go further the pluripotent state. So one of the main points that I would like to make it very clear, maybe it was not, is that once we have a bank of pluripotent stem cells from several animals from a species, we can provide other very more complex and further studies on the differentiation of the cells, and not we cannot think only on the use of that cells that we are reprogramming. They are just going and coming back for that same animal. It's a very important application for medicine, for regenerative therapies, for autologous therapies, but for any other applications, in special when we think about animal cell lines, when we have a biobank, and it's almost forever the cells, because they are very self-renewable, and we do not have to come again from scratch. Can do, for example, the reprogramming for one time and then do hundreds of other studies with these lines that were already generated.
Right, thank you. That's very helpful context. I think it was less about genetic level differences and more about maybe species level.
Oh, yeah, okay. So, well, if you want some specific pluripotent stem cells from that species, here in the lab, we always go first, we use one protocol that is robust for most every species. And when we see it's not working, we start changing small things. It's not always from the scratch. So we always have this protocol of reprogramming. It's very robust, at least between mammals. So it's never from really the scratch. So we always know how to handle which are the transcription factors we have to use, which are the supplements that we have to use. Maybe it's more on that side, the question.
Thank you. One question from the audience is what the possibility of genetic drift is when you're reprogramming cells and immortalizing them.
When we reprogram cells, we do not immortalize them. Well, people always ask me about immortalized cells, and I'm a little bit against them, because either they have a mutation, a spontaneous mutation, or they are genetically edited for being immortalized. So, or it is spontaneous or it is induced, and either way, I do not believe that it's something that we control much. And in the case, if they are genetically edited, they will not be very much acceptable for applications. About reprogramming, I'm not sure about what the question about genetic drifts. What would be that? I'm not sure.
I'll invite CISO to kind of add a comment in the question and answer section if they want to clarify. My understanding of it is just whether we can expect with the reprogramming process over time to see genetic variations kind of emerge. So
it's very common. It's very common. So chromosomal abnormalities, they are very common, and in every stem cell, in special for those who replicate a lot. So both embryonic and also induced pluripotent stem cells, since they replicate a lot, they have checkpoints for the cell divisions that are not the same as the adult cells, the somatic cells. So they are much more prone to present chromosomal abnormalities during culture. So we have to check them very often to see whether these abnormalities, if they appear, and that's an interesting question, because what we do to avoid losing these cell lines during times at the beginning in lower passages, we freeze them, we biobank a lot of them. So as one vial goes further, and actually at one time and maybe 10 passages, maybe 100 passages, they appear with chromosomal abnormality, then we go back and thaw a new vial from that same genetic background. So that's one way to avoid this, but well, it's normal to have a lot of chromosomal instability in the cell lines.
Thank you. I think that answered the question. Given the expression of the same pluripotency transcription factors between embryonic stem cells and iPSCs, why do you think the pluripotency capacity is higher in iPSCs? And why do you think embryonic stem cells lose pluripotency capacity or reach certain senescence in cultures?
That's a difficult question because, well, first of all, we induce the pluripotency with some factors that may be present or not in pluripotent stem cells. These ones, they are present, they are OCT4, OX2, semikala4, but we can also induce pluripotence by just changing the environment, the media, for example, and supplementing the media with some epigenetic modulators, such as enzymes and other substances. So the question about the same, the part of the question about the same pluripotence factors and why one is more pluripotent than the other, it is very questionable. And I would go for the answer that when we have different iPSC cell lines, we may have different pluripotent stages for each. They may be different between each other because some of them may be completely reprogrammed and some of them may not be completely reprogrammed. And what causes one to be more or less pluripotent than an embryonic stem cell lineage, it's not for me to answer that yet. I do not think we have the knowledge yet to decipher the pluripotency so finally, so specifically to know how to deal with different stages of pluripotency between cell lines and between iPSs and embryonic cell lines. So I rather not go further on the discussion because I just believe that each cell line that we have, we prove it is pluripotent, but we know that they are different between each one. And I'm not saying between animals, even between when we get one animal and different iPS cell lines that came from one primary cell, they are different between each other. So that's a very tricky question. And the second part of the question, which was, I forgot. Well, I think it was this part of the pluripotence.
Yes, the second part of the question was, why do you think embryonic stem cells lose pluripotency capacity or reach senescence?
If you are talking about farm animals, domestic animals derived pluripotent embryonic stem cells, I do believe that we do not know the exact factors that we would need to supplement our media for them to maintain pluripotency, but more than that, maybe this real pluripotent state that we isolate the cells from mouse, which is the inner cell mass, or from humans, which is the epiblast stage. Maybe in other species, it can be earlier or later, and we haven't really, we do not really know the exact time that the cells are prone to be isolated in culture. And when we do that, even there are experiments that have isolated them in different embryonic stages, and when they go to the plate, they lose pluripotency. So something is missing, obviously. And there is one research group, Professor Ross group. He was able to maintain embryonic stem cells from cattle from several passages and maintaining pluripotency. And but it has not been very much replicated yet, so it's still early to be so assertive that they have really found the pathway to inhibit 10-month pluripotency in cattle. But there are several groups that have made great discoveries and advances in this field lately. But actually now it's so much easier to produce the iPSC. So
Great, thank you so much. I will ask maybe one more question here before our final question. This one comes with a lot of background that goes above my head. So the expression of pluripotent transcription factors can be induced by viral vector-mediated transduction, such as retrovirus or lentivirus transduction, or through virus-free approaches like polypromoter vector or small molecule-mediated expression. iPSCs can reach immortality through epigenetic changes and upregulation of the enzyme telomerase, which are helpful for cell expansion and large-scale production. So the question for you is, which of these approaches do you think would be more appropriate for obtaining a genetically modified food instead of a transgenic one? And do these cells present stability in scale-up processes?
So I'll start from the end. So I was able to find the question here. It's easier for me to follow. So do the cells present stability, scale-up processes? I do not know. We have not scaled up our cells yet. The expression of preventative can be induced by vectors such as retrovirus. We use lentivirus. They work very well. We are trying to not use them anymore. Virus-free approaches, we use episomal vectors as virus-free approaches. They are now working well. Can reach mortality through epigenetic changes and upregulation of the enzyme telomerase. You know that some iPS cell lines, they do not reactivate telomerase. It's an important thing to think about. And even Dolly, which was a cloned sheep, she was not. She did not have her telomerase reactivated. But it is really helpful for cell expansion large scale. Which of these approaches would be more appropriate to obtain? When we use the lentivirus, the cell is already transgenic. If we want these cells for food predictions, I would not use a transgenic cells. If you want to produce food which is gene edited, it's different from a transgenic cell. So finally getting to the point here. If you want a modified food, I would go for a non-transgenic reprogramming of gene-edited primary lines. So you get your primary cell lines, you can edit them, and you can reprogram them. Or you can reprogram the cells and make your iPSCs, and then you can do the CRISPR or other gene editing tools in the iPSCs. This would be an easier way to go to the final genetically engineered product, but without being transgenic. Even though, because when we insert our transcription factors with an integrative vector, as lentivirus, for example, these exogenous factors that we are introducing, they can, for example, be reactivated, and then instead of one copy of the c-MYC gene, which is an oncogene, you can have two being reactivated. So these cells will always be more prone to be not acceptable for both regenerative medicine or for food as a final product. And well, again, I cannot say how scalable, how stable they will be when we put them in a large scale or like a bioreactor. I believe they will be fine if they are fully reprogrammed.
Thank you for your recommendations and for this entire presentation. There's just been so much enthusiasm in the chat for your great work and your generosity and kind of sharing what you know. Obviously, this field is still very nascent. We're in our early years and there's a lot of groundwork to lay. So we appreciate you for pioneering these methods that might lead us into the future for a more sustainable food system. I want to wrap by asking you one final question, which is, you have a captive audience of alternative protein scientists from around the globe. Is there anything you would like to leave this community with before we wrap today?
Yes, for sure. Maybe lots of things, but I believe that one most important, I would say it's a take-home message that I would like to share is that we are all here for a common goal. And although we need to be very stringent with technologies and to have control of everything and retroactability and everything, maybe at least when we in this side we are working with the basic sciences mostly, we have to be very open for new technologies and new possibilities. And I do know that some of the audience, at least part of the audience here, may be thinking, oh, this will never reach my plate. But 40 years ago, no one talked about IVF, in vitro fertilization. And 45 years ago, it was the first in vitro fertilization procedure. And today, we have billions of people that are born due to in vitro fertilization. So what we see as maybe a very important challenge at this moment, and maybe something that is not at our time of, it's not very practical. We need to think that maybe in 40 years or in 50 years, it will be a reality. So we have to try and to make things work today, and have to, maybe we will get some good answers and maybe we will not, and it's part of the process. We have to try and to believe in a better world.
I love that. Yes, the case for rational and impatient optimism, and just general open-mindedness, because we really don't know what the future holds, and our job here is to kind of explore the possibilities. I so appreciate what you've brought to the table today, Dr. Bressan. Thank you for your time and your perspective.
Thank you very much. It was
Thank you to everyone
a pleasure to be here today. Thank you
for attending. Yeah, it was a pleasure for me too. I hope everyone has a fantastic rest of their day, and do be sure to join us again next month for our monthly Science of Alt Protein seminar. Thank you so much, everyone. Take care.